Hello everyone, my name is Darmastuti. In this video, I will explain about seeing the shape of bacteria by simple staining and negative staining on bacteria. There are millions of organisms on Earth. Bacteria are one example of microscopic organism. It is a prokaryotic organism that has a very big role in life on Earth. Some are the bacteria cause a disease and infection, but some are beneficial to life. The structure of a bacterial cell is relatively simple, without cell nucleus, cell skeleton, and other organelles such as mitochondria and chloroplast. There are many ways to classify the bacteria, starting from the morphology to the tools of motion. Based on the shape or morphology, bacteria can be divided into coccus, bacillus, and spirillum. Meanwhile, according to the number of flagellum they have, bacteria are divided into altric, monotric, plevotric, and petritric. So, the question is how to determine the shape and type of bacteria? To identify the bacteria, we can use the bacterial staining technique. In this study, we will use simple staining and negative staining. Part 1. Methodology of simple staining. First of all, we must do sterilization. In this step, sterilizing the glass object using a methylate burner. The purpose of sterilization is to kill all forms of living microorganisms, including spores, in sterilized equipment. After that, we can drop natrium chloride onto the surface of the glass object and taking bacteria culture. After taking the bacteria culture, immediately place it on the preparation until it is even and wait for the preparation to dry. After drying, we can drop off safranin. The time interval between dropping safranin and rinsing with distilled water is 2 minutes. If it has been rinsed, it can be dried using a tissue. The last step on simple staining is observe the shape of the bacteria under microscope. Part 2. Methodology of gram staining. First, sterilizing the glass object using a methylate burner. Second, one end of the side of glass object is draped with eosin and taking a pure culture of bacteria. Third, glass object is placed at a 45-degree angle, dry it, and drop the immersion oil. Last, observe the bacteria under microscope. Simple staining is used to observe the shape of bacteria, whereas gram staining aims to classify bacteria into negative bacteria and positive bacteria. From simple staining, it can be concluded that the bacteria culture have a bacillus shape. Gram-positive bacteria are able to retain the main color because the cell wall of the group of bacteria are composed of mostly peptidoglycan, which are able to bind color and are not damaged when washed with alcohol. Gram-negative bacteria have a cell wall composition which is mostly composed of a lipid layer, so that when staining is less able to retain the color. As a result, the group of bacteria give a red appearance. From this observation, it can be concluded that bacterial staining can be used to determine the shape and morphology of bacteria. This bacterial staining can also be used to classify bacteria into gram-positive and gram-negative bacteria. Gram-positive becomes blue or purple under microscope, while gram-negative bacteria will turn red or pink.